I'm now going to talk about something that looks incredibly complicated but happens a lot. So one of the things that we are doing now in our lab, we've reported on one of these actually today, is if a woman becomes pregnant and wants to know whether or not the child is going to be affected, you can take, at about 8 to 13 weeks, you can take a small bit of what's called the placenta that comes from the baby. And this is just an illustration of this technique. So this technique involves putting a particular kind of catheter in, which is just a technique which takes a tiny snip about the top of the head of a pig, about the size of the head of a pig. And it takes a little bit of what's called the placenta. It's called the chorionic villus, but that doesn't matter what it's called. It takes a bit, and then that comes to our lab. And we can look in that pinhead piece of material, we can look at whether or not that genetic defect is there. Now clearly, if people want to go ahead with that technique, which has a slight risk, and the risk is about 1 in 100 that the pregnancy will stop, then people, if the child is likely to be very severely affected, people would probably want to go ahead with a termination of pregnancy. The reason why this technique has been developed, and it hasn't been developed specifically for mitochondrial disease, this is the sort of technique that's often used in terms of many other genetic conditions, things like Down syndrome and other conditions. This technique is used a lot, and it's only starting to be used for mitochondrial conditions. I think, and I'm looking at Emma at the back there that does some of the analysis here, I think we've done about 7 or 8 of these now, and we report back from patients who have come from all over the country. These aren't patients that are all biopsied here. Another technique, which is done very similar, is just getting some of the fluid. The reason why this technique is less popular is that this is done much later in pregnancy, and obviously the complications of termination at this stage are much more difficult. Okay, so this is something that we can offer families now. Again, it's something that is relevant to some families, not relevant to others. And it's one of the things why you need to have specific information given. But what else can we do? Can we be clever about this? Can we stop the fact that if you have the genetic defect, that the woman would need to have a termination? Well, yes, we can be clever. And yes, we are being clever by using a technique that's been developed up in labs around the country. And this is a technique where the family goes through IVF. But I was talking to people about whether or not I need to explain IVF. I think you all know about test tube babies. So, in other words, this is fertilization outside of the body, and what happens is the egg gets fertilized, and what happens is it develops up as an early embryo. And what you can see here is that this egg, this fertilized egg, becomes an embryo, and you can see here it's got lots of cells. And what we can do now is, down a microscope, we can take one of those cells and we can analyze one of those cells to see if the genetic defect is present. That means that then we would only put back the embryos which don't have the genetic defect or have less risk of the child being affected. Now, this was something that couldn't be done anywhere in the UK until July last year, and we're the only centre in the UK that's offering this. And again, this is very, very specific for individual families and individual couples, and something that we would discuss openly with people who want to discuss this. So what else is new? Well, this just comes back to some of the kind of research that we've been doing, and what I'm actually going to do is actually showing you a clip from the BBC News. You're going to say, well, why show a clip from the BBC News? I'm actually going to show this clip because it's about work that came from Newcastle, and I said to my wife, because I couldn't bear to watch it, I said to my wife, well, what was it like when it came on the telly? She said, it was the first time I ever really understood what you did. So,
Good evening and welcome to the BBC's News at 6. British scientists have developed a pioneering technique that promises new hope to families that live in the shadow of a range of inherited diseases. A team at Newcastle University have shown it's possible to create a human embryo using genetic material from 3 people, the mother, the father and another woman. Crucially, the child will have all the characteristics of the parents, but without the DNA that carries the inherited disease. Here's our medical correspondent, Fergus Walsh.
A newly fertilised human egg being manipulated in a lab, a microscopic image that has become familiar, but you are looking at something radically different, a new frontier in genetics. Because these scientists in Newcastle believe they can actually fix a genetic fault which has caused illness, death and heartache for families down the generations. Like for Edward and his mother. He's severely disabled with a host of health problems, the result of a DNA fault passed on by his mum Sharon. The same genetic illness meant she endured the death of her 6 other children, all within days of birth.
You can't imagine it happened once, but happened 3, 4, 5, 6 times. And then to be told, obviously my son's got something. It's a disease that causes Edward to have chronic pain in his muscles, which makes him scream throughout the night. He doesn't walk now, and he needs full-time looking after.
The disease is found in these structures, mitochondria. They're the power packs for each cell and have their own DNA. They're passed on through the woman's egg. This research used newly fertilised eggs donated after IVF. The nuclei of the sperm and egg contain all the crucial genes from both parents. These were removed, leaving behind the mother's mitochondria. The nuclei were transferred to another woman's egg, carrying its own mitochondria. The embryo had the parent's genes plus a tiny bit of DNA from a second woman.
all we're doing is changing over the power supply. All the key information that makes a person or a baby is going to be there. And all we've done is correct this problem with the power supply.
None of the 80 embryos they produced was implanted in a woman, but the Newcastle team think they'll be ready to treat couples in about 3 years. But critics say that must never happen.
Parents are seeking to have a child of their own, but what might eventually happen is that they will have to share this child with a third person. In other words, the person providing the egg.
I do it.
Sharon knows this research comes too late for her, and it would take a ruling from the Health Secretary before the treatment could be used to help around 1,000 other women with the same condition to have a healthy child.
And Fergus joins me now. So what next, Fergus? When will the first family benefit?
Well, George, this research in the journal Nature shows that British scientists are once again at the cutting edge of genetic research. Now, if they can demonstrate through animal trials and further lab research that this looks like it's safe, I think it's very likely that this will eventually be allowed. Let's deal with this issue of DNA from 3 different adults. The donor DNA from the second woman will be 37 genes found only in the mitochondria. Crucially, they won't pass on any human attributes. Contrast that with 23,000 from the parents. Now, critics will worry about the child's sense of identity, having this second, albeit small, maternal DNA source.